Thank you so much. I'm Ghazaleh Soleimani, and I'm recently a postdoc in a joint program of Amir Kabir University of Technology and Iranian National Center for Addiction Studies. First of all, I want to say thank you for organizing this fruitful symposium, and I'm really happy that we are here together to talk about recent advancements in neuroscience. In the next 15 minutes, I'll be talking about one of our recent research in the field of non-invasive brain stimulation technologies and substance use disorders. As a brief introduction about different types of neuromodulation methods, I can say that the use of electricity, as you know, is as a therapeutic medical application is not a recent discovery, and it dates back to ancient Egypt. They were familiar with an ability in catfishes to produce electricity, and they used this electricity for headache and migraine, for example. And after that, with the advancements of technology, different types of neuromodulation methods were emerged. And one of them was transcranial electrical stimulation. And in this study, I want to focus on a specific type of TS method, which is named transcranial direct current stimulation, or TDCS. As you can see in this slide, TDCS has a really simple structure, something like a 9-volt battery, and a small a direct current about 1 to 4 milliamp, for example, passes through the scalp into the brain based on placing at least 2 electrodes as anode and cathode, and at least one of them should be placed over the scalp. And after that, you hopefully can modulate your brain. And there are lots of different evidence that reported that TDCS can affect different types of neuropsychiatric disease such as depression, migraine, obesity, and substance use disorders. We were interested in the field of addiction medicine, and one of our recent research in the field of one of our previous systematic review for the application of transcranial electrical and magnetic stimulation in the field of addiction medicine showed that TS and TMS methods can be effective in the field of substance use disorders, but the main problem here is that we are not familiar with the neural substrate of these types of technologies in substance use disorders, and more research is needed to find the mechanism of action. So in order to study the mechanism of action for TDCS in substance use disorders, we conducted a randomized double-blind sham-controlled clinical trial with a crossover design, and 15 participants with methamphetamine use disorders were randomly assigned to receive active or sham stimulation with at least 1 week washout period. And in order to apply TDCS, large electrode pads were placed over F4 and F3 locations in EEG standard system for bilateral DLPFC stimulation. And during the active session, people received 20 minutes of 2 milliamp stimulation, but during the sham session, people only received 30 seconds of TDCS. And in order to study the brain functions, MRI data, including structural MRI, resting state fMRI, and task-based fMRI, were collected before and after active and sham stimulation. And behavioral data, such as craving score, were also collected immediately before and after MRI session. During the resting state, people only look at a cross inside the scanner, but during the task-based fMRI, we designed a standard drug cue reactivity task, and people were exposed to drug versus neutral cues, something like these pictures. As you can see, for example, you can see similar content in both pictures. For example, you can see hand inside the pictures, but one of them is related to drug and the other one is related to neutral cues. And we wanted to determine the effects of TDCS while people are exposed to drug versus neutral cues. And because the effects of TDCS, especially with large electrode pads, are not restricted to a single anatomical region, and because different parts of the brain are communicating with each other, we were interested to analyze our data at the network level. And we focused on 3 main networks that showed abnormalities in people with substance use disorders. One of them was default mode network with two main nodes, including ventromedial prefrontal cortex and posterior cingulate cortex. And we know that DMN is activated during retrospective processing. And the other network was an executive control network with two main nodes, including dorsolateral prefrontal cortex and posterior parietal cortex. And we know that ECN is activated during, for example, cognitive processing. And previous neuroimaging studies showed that there is a kind of anti-correlation between DMN and ECN. For example, highly activated or connected DMN will have a lower activation in executive control network. And we also considered a third network, which is named ventral attention network, with the main nodes, including insula and dorsal anterior cingulate cortex. Ventral attention network works as a gear inside the brain and determines which network, including DMN and ECN, should be activated in each moment. And previous studies showed that people with substance use disorders compared to healthy subjects have a highly activated or connected DMN, and because of the anti-correlation between DMN and ECN, the activity and connectivity inside the ECN is significantly lower in people with substance use disorders. And the main hypothesis for the application of TDCS over the DLPFC is that when we apply the current over DLPFC, we help to ECN, we modulate ECN, and hopefully the activity and connectivity inside the ECN will be increased. And we will have more connectivity between ECN and ventral attention network. And because of the anti-correlation between ECN and DMN, we hopefully can decrease the connectivity or activity inside the default mode network. And we wanted to check this hypothesis while people are exposed to drug cues. So we consider, we focused on ECN, DMN, and ventral attention network. And in the first step, we created computational head models for all of 15 participants in our study, and we extracted electric field strength from ECN, DMN, and ventral attention network nodes. And we found that, as we expected, PFC nodes in ECN, DMN, and ventral attention network receive the highest electric fields inside the brain because they are the nearest node into the stimulating electrodes. And we considered these three nodes as the seed region for further seed to whole brain analysis. And we use a specific type of task-based functional connectivity analysis, which is named generalized psychophysiological interaction or context-dependent correlation analysis. And we found that we searched for brain regions that showed time by intervention interaction, significant time by intervention interaction. By time, I mean post versus pre, and by intervention, I mean active versus sham. And we found that the PFC node in the ECN and DMN changed their connectivity with two main clusters inside the brain. And our post-hoc analysis also showed that the direction of changes are different in ECN and DMN. For example, during the active stimulation, the connectivity between ECN and the significant cluster was increased when we considered the PFC node in ECN, but active stimulation decreased the connectivity in situ whole brain analysis when we consider the PFC node in the DMN. And in the next step, we also performed region of interest to region of interest analysis to determine task-based changes in task-based connectivity, and we considered all of the main nodes in DMN, ECN, and ventral attention network. And we found that PPI connectivity increased significantly within and between ECN and ventral attention network, but all of the connectivity within the DMN or between DMN and ECN were decreased, and we only found one decreased connectivity inside the ventral attention network. So it seems that our first hypothesis about the effects of TDCS to increase the connectivity inside the ECN and ventral attention and between ECN and ventral attention network and decreased connectivity inside the DMN network is correct. And we also found some significant correlation between electric fields and changes in task-based connectivity inside the brain. In the next step, we also checked this hypothesis based on functional activity rather than functional connectivity, and we found that functional activity inside the PFC node in ECN and ventral attention network were increased after active TDCS, but the functional activity was decreased in default mode network. But now a question was popped out: if all of the PFC nodes in ECN, DMN, and ventral attention network received a strong electric field, why we have a different behavior in these networks. In the next step, we calculated the direction of the electric fields inside the PFC nodes. And interestingly, we found that we have outward electric fields in the PFC node in DMN. It means a negative direction in the PFC node in default mode network, but the direction of the electric fields inside the ECN and ventral attention network was inward. And we also found some correlation between tangential and normal components of the electric fields inside the DMN and changes in functional activity inside the brain. And when we calculated the craving score, we also found when we compared after versus before TDCS, we found that TDCS decreased craving score significantly only when people received active stimulation, and craving score was not changed significantly when they received sham stimulation. And we also found some significant or nearly significant correlation between changes in craving score and changes in task-based functional connectivity. And if I want to have some conclusion from our study, first of all, we have the number of subjects. We need more subjects in future studies to make sure that our hypothesis is true. But our results generally give an insight into how bilateral DLPFC stimulation can modulate task-based functional connectivity at the network level. And we also provided a preliminary evidence for the importance of network-level connectivity during a standard Curie activity task for people with substance use disorders. And we also suggest that for future studies, it might be better to study the effects of TDCS at the network level. And it would be really great if we can create computational head models for all of the participants, and not only electric field strength, but also the direction of the electric fields might be informative for our future studies. And finally, thank you for your attention. I want to say thank you to my supervisor, Dr. Hamed Akhtiyari and Dr. Farzad Touhidkha. Many thanks.
Thank you very much. It's very on time. We have time for a few questions. Dr. Ghazaleh, please.
Hello. Can you put your chat back? It was I want to ask you a question about the paradigm.
Sure.
So can you bring the paradigm page, the initial, right, 6. Okay. So this was a good study. Thank you. So can you explain the paradigm one more time?
Sure. We recruited 15 participants with methamphetamine use disorders, and people were randomly assigned to receive active or sham stimulation with at least 1 week washout period. And for applying TDCS, we placed two large electrode pads over F4 and F3 locations in EEG standard system. During the active stimulation, people received 20 minutes of 2 milliamp stimulation, but during the sham session, people only received 30 seconds of 2 milliamp stimulation. And in order to study the brain functions, MRI data were collected immediately before and after each session, both active and sham stimulation. And in order to determine the craving score, we also collected behavioral data immediately before and after each MRI session.
So,
during resting,
So for craving, was it just based on questionnaires? How did you measure?
Yes.
And what?
Yes, VAS score. We collected VAS scores.
So I mean, what was? Can you give examples of what kind of questions they answered?
Yes, and when people placed inside the scanner, they are exposed to met versus neutral cues. So we think that after each MRI session, they may have a higher craving score. In each questionnaire, we ask, for example, how much craving do you have right now, and based on a Likert scale, they choose an answer for this question.
I see. So in each fMRI session, they saw both a mixture of methamphetamine and neutral cues, and you asked for their craving before and after the MRI. Is that right?
Yes, yes.
I see. And where the stimulus battery that you used, is it something that is publicly available, or is it something that you guys made?
Yes, I think our TDCS device was Neurocan, I think.
No, not the TDCS.
Um,
The pictures, the cues.
yes. I think I'm not sure about these pictures, but for this data was collected some years ago. But today we have a standard database for drug versus neutral cues that is freely available, and they are standard. For example, about they should have similar content, for example, and we now have a standard database that is available online. But
But
this database, I'm not.
this database was not using the standard, was it? Who collected this
Uh,
data? Is it Dr. Ikhtiyari's data? He collected that, or you collected this data?
No, I didn't collect this data, and the database is a standard, but I'm not sure about freely available, if it is freely available. We have published this database based on resting state fMRI before in Human Brain Mapping Journal, a person who names Dr. I'm not sure, Dr. Shah Babai collected this database, I think.
Okay. So, and the craving pre and post, when you mentioned craving pre and post, is basically right after the TDCS stimulation, post TDCS and pre TDCS in the same day, right? In the same day.
This icon, I'm not sure if you can
Yeah, I
see.
can see
Let
this kind
me.
of questionnaire you asked before and after how much craving.
Yes, this icon is an indicator of behavioral data, and a craving score was collected in all of these time points.
Yes.
But our
Right.
data, our results showed that only when we couldn't find any time by group interaction, any significant time by intervention interaction. But when we compared after to before stimulation, we found significant decrease in craving scores.
Um, yes, but my question was that the stimulation, I mean, the after and before is in the same day, right? Same day. So you do a craving and then you do stimulation and then you measure craving again after. And this happens in the same day, right?
Yes, if I go back to this slide, um, this line is
Yeah, the washout period is 1 week, but the thing pre and post is in the same day, right? Pre TDCS and after TDCS is in the same day.
yes, I think, but I'm a little bit confused. I'm not sure, but I think it's so.
Okay. You say that this study is published?
Yes, the database and this study for resting state of MRI at the network level was published,
Uh-huh.
but in this study we are working on task-based MRI.
Right, but it's the same data set. Would you say who was the first author of that? Uh, so I can check.
Previous study,
Can you just study? You say the same study has been published before, right? This study has been published before. You're just analyzing some other parts of it, right?
yes, the resting state of MRI data was published by, and the first author was Dr. Ali Reza Shahbabai, I think.
Thank you, thank you. Okay.
And I can share the link in the chat box if it's
Yeah,
necessary.
if you can do that, that would be perfect. Thank you.
You're welcome.
Hey, thank you, everyone.